Welcome to Bigfoot Forensics. Today we're going to embark on the fascinating journey into the intricate world of DNA and trace evidence. This journey will take us through the microscopic realms of hair, DNA, and other minute particles that play a critical role in forensic science. So let's get to it. What did the DNA say to the other DNA? Do these genes make me look fat? I suck at telling jokes. I'm sorry. I thought I'd give it a try, but maybe I'll just stick to this. First, let's define trace evidence. Trace evidence refers to tiny particles that are sometimes invisible to the naked eye. Examples include hair, pollen, plants, geological materials, gunpowder, wood, cosmetics, clothing, fibers, glass, paint chips, plastic fragments, and other types of micro debris. It's important to note that DNA is generally excluded from the term trace evidence. However, this is where it can get confusing. We often hear the terms trace DNA and touch DNA used interchangeably. But trace DNA is the proper term. We say trace because we're referring to very small amounts. Using the term touch DNA can wrongly imply that DNA got there by being touched by the original contributor. We now understand that DNA is easily transferred from surface to surface with the slightest contact. For example, you and I shake hands, I touch a safe at a burglary, both our DNA is most likely going to be deposited. The quantity of DNA evidence can be very low, sometimes just a few cells, but those cells can hold significant forensic value, and they can still be used to identify you, even though I'm the one that touched the safe. Luckily, DNA analysis is so sensitive now that we're looking at who's the top contributor, who left the most. So let's talk about hair DNA. When it comes to hair DNA, the specifics are equally fascinating. For instance, a complete hair sample with the root is necessary for a paternity test. We need more information. In forensic investigations, even a cut hair sample without the root or bulb can be useful. However, the procedure to analyze it's a lot more complex. Quick bit of trivia. The root is what lies below the surface of the skin. It's made up of many different parts, including the bulb. The follicle, however, is a tube-like structure. It's like a pore that surrounds the root and the strand of the hair. So these are the two different types of DNA from hair we're going to talk about. The first is mitochondrial DNA. This type of DNA can be extracted from hair without the root. It's inherited from our mothers, and it doesn't undergo recombination, making it identical among maternal relatives. The second is nuclear DNA. Found in the nucleus of cells, nuclear DNA is inherited from both parents, and it does undergo recombination. If you're not familiar with what recombination is, think about it this way. You have your mother's DNA, you have your father's DNA, coming together to make you. It needs to combine together as one. Some traits you get from your father, some you get from your mother. Simplified, but you get the idea. If you're wondering why the hair you collected that you're absolutely sure is from a Bigfoot isn't coming back to anything, here's a possibility. Collecting DNA from hair without the root can be challenging, particularly in identifying species. The best outcome might sometimes be of unknown origin. Several factors can affect the integrity of DNA samples. One is water exposure. Now remember, we're outside collecting DNA. Water, sun, here we go. So water exposure. Washing hair can degrade DNA by breaking down the phosphodiester bonds. Dye and other beauty chemical treatments can alter the hair cuticle and degrade DNA as well. Another big one is the sun, and it's a really big one. Exposure. UV light degrades DNA samples. Think about this when collecting hair from the outdoors. Time can be an issue just because of the sun's UV rays, as well as the rain. Oh, and that reminds me, why did the DNA sit in the sun? It wanted to unwind. Okay. All right, other factors include the age of the sample, collection methods, storage conditions, bacteria, exposure to other chemicals and temperature extremes. So let's dive into eDNA. We keep hearing about eDNA in this field, correct? All right, so environmental DNA or eDNA. It's another fascinating topic, okay? So eDNA is either nuclear or mitochondrial DNA released from an organism into the environment. It can be detected in water, soil, or sediment. In water, eDNA lasts about 7 to 21 days, while in the soil or sediment, it can persist for years or even decades. This also means a more contaminated sample, because this sample could contain DNA from a lot of different animals from the area the sample was collected. This persistence makes eDNA a powerful tool in environmental monitoring and conservation biology. All right, so for those of you interested in the practical applications, I made you a lengthy list of some tips for eDNA collection. It's listed below and labeled Study Guide for Collecting eDNA from Soil. I also made one for collecting swabs, and it's also below. All right, now the bigger problem. Where are you going to send it? Honestly, I have no clue. But this is what you should do with it until you find out. The survival of DNA on a swab can depend on several factors, including the storage conditions and the initial quality of the sample. Generally, here are some guidelines. All right, let's talk about short-term survival. Room temperature. DNA on a swab can survive for a few days to a few weeks at room temperature if kept dry and away from direct sunlight. However, the quality of the DNA may degrade over time due to environmental factors. Long-term survival. Refrigerated. 35.6 to 46.4 Fahrenheit. That's doable with a regular refrigerator. So DNA on a swab can remain viable for several months to a few years stored this way. This is a common practice for short-term to medium-term storage. All right, a little bit longer, frozen. -4 or lower. We're talking Fahrenheit, -4 degrees Fahrenheit or lower. For long-term storage, freezing the swab can preserve DNA for many years. Properly frozen DNA can remain stable and usable for decades. It's important to avoid repeated freeze-thaw cycles, though. That can degrade the DNA. So what are some factors affecting DNA stability? Let's go with moisture. We've been talking about moisture. Moisture can cause mold and bacterial growth, leading to DNA degradation. Ensuring samples are dry before storage is crucial. Contamination. Proper handling and using sterile tools are essential to prevent contamination from other biological materials. Your storage containers, using breathable containers, example, paper, envelopes instead of plastic, can prevent moisture buildup. All right, some best practices. Immediate drying. Moisture is your enemy. Immediate drying. Ensure that the swabbed sample is dried completely before storing it. Secure and controlled environment. Store samples in a controlled environment with restricted access to maintain the integrity of the evidence. We're talking chain of custody now. You got to be able to prove it wasn't contaminated. By following these guidelines, the DNA collected on swabs can remain viable for analysis for an extended period. So when collecting your samples, you're going to need sterile and DNA-free tools, swabs, or whatever you're using. Okay, now we're going to talk about safety and practical considerations. I just want to get some stuff clear. If we're talking about collecting DNA, I also want to make sure that you're safe and make sure the DNA is safe. So a while back, people were out there saying that you could boil your stainless steel instruments to kill any DNA or, because you can boil water for drinking. All right. So we're talking about DNA, not necessarily killing off viruses and bacteria. So here are some things that you need to know. Boiling water can degrade DNA, but it may not completely destroy it. So DNA is relatively stable at high temperatures, but boiling, it can cause the DNA strands to denature and break down over time. That's over time. For complete destruction of DNA, higher temperatures and longer exposure times are required, often achieved through methods such as autoclaving, which involves using steam under pressure to reach those temperatures that are above boiling. So boiling, it's not going to kill a lot of the bacterias. When some big ones are tetanus, gangrene, anthrax. So just keep in mind that it's not going to kill everything. And it's going to take a much longer time than you originally thought to kill that DNA if you're trying to make sterile tools. So what are some other methods? Unfortunately, still a lot of people think that rubbing alcohol is going to kill stuff because it kills the germs and viruses, and it's one of the bigger things that they put in your hand sanitizers, right? Okay, so let's get this clear. Rubbing alcohol does not kill DNA. Alcohol is actually used in isolating DNA, and it helps preserve it. However, rubbing alcohol will kill bacteria, most viruses, and certain fungi. So using an autoclave. Autoclaving, sometimes called steam sterilization, is the use of pressurized steam to kill infectious agents and denature proteins. This kind of wet heat is considered the most dependable method of sterilizing laboratory equipment and decontaminating biohazardous waste. But there's one that beats everyone out, and that's using bleach. A 10% solution of bleach to water. Contact time, 5 to 10 minutes, is generally sufficient to degrade DNA effectively and kill anything else. Okay, so don't forget to rinse after your contact time. It's important to thoroughly rinse the tools or surfaces with water to remove any residual bleach, which can be corrosive to certain materials and might interfere with subsequent analyses. And don't forget to use a fresh solution each time. Always prepare a fresh bleach solution before each use, as bleach can lose its potency over time. I couldn't talk about bleach if I didn't tell you to use gloves and eye protection and ensure proper ventilation when handling bleach to avoid skin and eye irritation and inhalation of the fumes. Okay, so what I'm going to do is I'm also going to provide some links down in the bottom of where you can go and get sterile tools. If you don't want to bother cleaning yours over and over and you want to open it new every time, I'll provide some websites too where you can just go and buy straight sterile gloves, tweezers, scalpels, anything you can think of, and you can one time use it and toss it. Honestly, that's the route that I prefer. Also on camera, I can show me opening that particular instrument and showing that it's being opened for the first time, and then there's no argument on whether it was sterile or not. Okay, so I guess the bottom line is DNA testing is highly sensitive and it can provide invaluable information. It also comes with challenges, especially when dealing with trace evidence and environmental samples. It's crucial to follow best practices for sample collection and storage to ensure the integrity of the DNA. If you enjoyed the show, don't forget to hit like and subscribe, leave a review with your feedback. If you have any questions or topics you'd like to hear about, just feel free to reach out. Let's continue to explore the wonders of forensic science together and have a great evening.